Welcome back to The Explainer. Today we're diving into something called biochemical cofolding. And trust me, it's a process that's completely rewriting the rules of medicine, allowing us to go after diseases we once thought were, well, totally untouchable.
So let's kick things off with a huge question, one that honestly kept scientists up at night for decades. How in the world do you drug something that's, well, undruggable? I mean, how do you hit a target that doesn't seem to have a bullseye? This isn't just some abstract puzzle, you guys. This is one of the biggest roadblocks in medicine.
So this whole mystery, it's all about a certain kind of protein. And according to everything we thought we knew about biochemistry, these things should have been impossible to target with a drug. Totally off limits.
All right, let's meet the main character in our story, a protein called MYC. Now, MYC is a major bad guy in a lot of human cancers. But for something like 30 years, it had this reputation: it was undruggable. And you're probably asking, why? Well, it's because MYC is what's called an intrinsically disordered protein. Think of it as floppy, shapeless, with no clear dock for a drug to park in. It was literally like trying to grab a fistful of smoke.
And yet, a drug worked. I mean, can you imagine? Researchers were completely floored when a new compound actually managed to shut MYC down. So what was the secret? The trick wasn't to go after MYC by itself. No, the breakthrough was targeting the interaction, that critical moment of connection, this kind of molecular handshake, when MYC finds its partner, Max, and they fold into shape together.
Okay, so to really get how this amazing breakthrough happened, we need to talk about the idea at the very heart of it all, cofolding. And believe me, this represents a massive shift in how we've always thought about the basic building blocks of life.
You see, for the longest time, the textbook picture of protein folding was kind of a solo act. You know, you have this long chain of amino acids, and it just twists and turns into its final 3D shape, all on its own. But it turns out nature is way more social than that. Cofolding? It's a team sport.
So, what is cofolding? To put it really simply, it's when two or more proteins don't fold by themselves. They fold together. Their journeys are completely linked up. The final shape of one protein literally, and I mean literally, depends on its partner being right there with it. They absolutely need each other to get into their final working shape.
Here's a great way to think about it. Origami, right? Folding one paper crane is pretty straightforward. But now, what if you had to fold two cranes at the same time and they had to share a wing? You just couldn't finish one without the other. That final intricate shape only works when both pieces come together as one.
And this leads us to something called the disorder paradox, which is just so cool. See, a lot of these proteins, like our friend MYC, are totally shapeless when they're alone. They're just kind of there. For years, this drove scientists crazy. They were thinking, how on earth can something with no fixed structure actually do anything useful in the cell?
And get this, this isn't some weird, rare thing that happens in a forgotten corner of biology. No way. It's super common. We're talking estimates of up to 30 to 40% of all the proteins in the human body having these disordered regions just waiting to cofold. This is a fundamental core principle of how we work.
So you might be asking, why would biology bother with this whole messy, disordered strategy? Well, it turns out there are some massive advantages. For one, you get promiscuity, and I mean that in a biochemical sense. One protein can bind to lots of different partners. You also get incredible regulation. These interactions can be turned up or down, just like a dimmer switch. And finally, speed. These floppy guys can find their partners way faster than rigid, structured proteins can, which is absolutely crucial for sending quick signals inside our cells.
Okay, so this molecular handshake, it's not just some neat little trick that happens in a petri dish. No, this process sits right at the crossroads of human health and, unfortunately, some of our most devastating diseases.
Because when this process goes wrong, the results can be catastrophic. Think about neurodegenerative diseases like Parkinson's or Alzheimer's. What's happening there? Well, proteins like alpha-synuclein and tau are essentially cofolding incorrectly. They're getting together and forming these toxic clumps that damage and kill our neurons. That friendly handshake, it turns into a death grip.
But what if we could control that handshake, right? I mean, if a faulty handshake is what's causing the disease, then maybe, just maybe, getting a handle on it could be the key to a cure. And that single thought, it opens up a whole new playbook for how we design drugs.
And this brings us right back to our MYC mystery and how they solved it. It's so brilliant. The drug doesn't even try to grab onto the shapeless MYC protein. Instead, it acts like a blocker. The drug molecule just parks itself right in the docking bay, physically getting in the way of where Max was supposed to connect. The handshake is blocked, the cancer-driving machine can't assemble, and just like that, the disease signal is shut off. Game over.
So now that we understand this principle, you can see why people are so excited. We are standing on the edge of a genuine revolution in drug design. This idea of targeting the interaction and not just the lone protein, it changes everything.
Think about this. For decades, just figuring out the 3D shape of a single protein was considered a massive, almost impossible challenge in biology. Then, boom, 2021 rolls around, and AI systems like AlphaFold basically solve it. So what's the next great frontier? You guessed it, predicting and modeling how proteins fold together.
There's this quote that I think just nails it. Biochemistry has always been about relationships. Cofolding just makes that literal. And that's really the big shift here, isn't it? We're moving past just looking at individual parts under a microscope. Now we understand that it's the relationships, the interactions that really matter in the bustling city that is a living cell.
So, what's the big takeaway from all this? It's that this massive universe of so-called undruggable proteins, well, it might not be so undruggable after all. By shifting our focus to the handshake, to that moment of cofolding, we've unlocked a completely new strategy. And that leaves us with a really exciting question to think about. Now that we know how to interrupt the handshake, what's the next undruggable target we can finally hit?